One of the exciting aspects of ASH over the past few years, and I'm glad to see it continuing, is the so-called clinical trials in progress section. And I really want to highlight all of the viewers out there to make sure you see these posters. So these are, by definition, either preclinical or very early stage clinical trials that have just started, maybe have not yet had a chance to have mature data, but it allows sponsors, academic investigators to get the word out there about these trials. And one of those trials that I'm helping to co-lead is this AML-01, which is the UCART CD123. The principle here is very important and new to some viewers. CAR T cells, a lot of people are getting familiar with, chimeric antigen T cell receptor therapy, but most of the ones that you're familiar with or that are approved are autologous. They come from the patient themselves. This study is among the first to investigate so-called allogeneic CAR T cells, or off the shelf. So they come from healthy random donors, and then they are put through the CAR T process. And so the early efforts now are underway in AML, which does not have any approved CAR T therapies, and this targets CD123, which we were able to investigate prior. So the drug tagraxofusp, which is approved for BPDCN, is the first and only CD123 targeting agent, but importantly, it's not approved for AML. So that's the premise: CD123 overexpression in AML, off-the-shelf allogeneic CAR T cells. And so the only public data to report so far is what's in the poster, which is this is an ongoing phase 1 dose escalation study with this very novel method, which includes the lymphodepletion, the infusion of the allogeneic T cells, and then watching for safety and early signals of efficacy after that. It is a multi-center study with my colleague, Dr. Gail Robos at Cornell, as the other co-leader, as the first author of this study, with me serving as the senior author. And I think in general, this field of off-the-shelf or allogeneic CAR T is important as we investigate it in other forms of leukemia, particularly in ALL, with my colleague, Dr. Nitin Jain, and others leading those efforts.